Metagenomic sequencing. It's also called deep metagenomic sequencing. Meta, I think, usually means around. So deep around the genome sequencing. So what is that? So the paper that set me off was from a journal called Quantitative Biology, which is an interesting name for a journal, since biology is not quantitative. But anyways, and it was published not so long ago, March 2020. Looks like a mixture of, Western Nathans and Jads with Ryans with some Asian people, not sure from where. And the title is Identifying Viruses from Metagenomic Data Using Deep Learning. So essentially what they're talking about is we all have heard about how they come up with the sequence that to claim they have sequenced the entire genome of such and such a virus. And it basically goes something like this. I know you've heard this all, but you take unpurified stuff. I sound like I'm on a broken record here. You do this culture. We all know what that is. The culture breaks down into all these pieces, and the genetic material that's in the results of the breakdown of the culture obviously came from many different sources. It came from the human being, if you're doing a human sample. It came from the bacteria that live in your lungs, or the human being they took the sample from. There's fungus, and there's all kinds of other cellular debris in that mixture. They then added it to fetal calf serum and monkey kidney cells. So now we have a whole nother line of breakdown products. And then they start chopping that up into little pieces. They discard the ones that they don't want, either DNA if they're looking for an RNA virus, or RNA if they're looking for a DNA virus. They discard the ones that they say are from humans or microbes, even though, truth be told, there's no way of actually distinguishing between the two. And then they start amplifying them with the PCR process, because you can't find these little pieces until you amplify them. Amplify is just a fancy word for doubling them, or a fancy word for making more of them. And then once you've got a whole bunch of sequences, you start piecing them together like letters or words, and you try to make paragraphs and chapters and books out of those little pieces. So that's the sort of normal, what's called unbiased, which is not unbiased at all, because you need to tell it what you're looking for. Otherwise, the rule of the world is you can't make something that's accurate rendition of anything out of little pieces unless you've seen an example of the thing you're trying to make. You can't reproduce a book with the exact letters and words and paragraphs and sentence unless you know the book, and then you could take the little words and letters and make them into the book. If you don't have the book, you can't do it. So that's why it's not unbiased. And unbiased de novo, meaning anew. So they're doing this for a new virus, unbiased de novo sequencing. So in metagenomic or deep metagenomic sequence, what they're doing is getting rid of the culture part. And so they just start with the sample, like the lung fluid, and then they essentially do a deep amplification. That's where the word deep comes in. Deep meaning they put I believe, the entire brew through 45 cycles, or at least the sequences they're looking for. That's probably not the entire brew. The sequences they're looking for through 45 cycles, and then they try to piece those together to see what they have. So that's what deep metagenomic sequencing is. So let's look at some of the background of this. First thing, the recent development of metagenomic sequencing makes it possible to massively sequence microbial genomes, including viral genomes, without the need for laboratory culture. Existing reference-based and gene homology-based methods are not efficient in identifying unknown viruses or short viral sequences from metagenomic data. In other words, they want to do away entirely with this whole business of taking the snot and growing it in a culture, even though that they claim is the only way you can prove the existence of a virus and prove that it causes disease. That's obviously too messy and takes too much time, and you have to kill a bunch of fetuses and keep monkey cells growing forever. And so they don't want to do that. They want to do away with the whole thing and just sequence. In other words, find what genetic sequences, what little pieces of genetic material are in the original sample. And then they're saying the reason we came up with this deep learning technique is the existing ways of doing this are not efficient in identifying unknown viruses, which means, I believe, that if you don't know what virus you're looking for, you can't find it with the current deep metagenomic techniques. So they want to find a new technique which will find unknown viruses without having to put a template in there, which is now 2 steps bizarre, because they don't even have a template. They just start sequencing and amplifying whatever's in there, and then somehow they're going to come up with the new virus. So what do? Well, here we developed a reference-free and alignment-free machine learning method called Deep Vir Finder for identifying viral sequences in metagenomic data using deep learning. In other words, they're not even anymore going to compare it to previously and fraudulently created sequences. They're not going to create it compare it to any sort of previously aligned and fraudulent sequences. They're just going to identify sequences by putting in primers and amplifying the whole thing or whatever they want and seeing what they get. So this is like seeing what we get. We're going to amplify whatever we want, or maybe the whole thing, and see what sequences are in there. So they did this, and here's what they said. They applying Deep Vir Finder to real human gut metagenomic samples. We identified 51,138 viral sequences belonging to 175 bins in patients with colorectal cancer. 10 bins were found associated with the cancer status, suggesting viruses may play a role in colorectal cancer. So what are they talking about here? So they took a bunch of people and took samples, presumably stool samples. Some of them had colorectal cancer. They started sequencing these and looking for these sequences. They found initially 51,000 different viral sequences. They belonged to 175 different bins in patient, that's categories, and 10 of these categories, or so-called viruses, were found more in people with cancer status. And since these new sequences are now being called viruses, that's the important point here. These little pieces of genetic material, which you have no idea, and I can't emphasize that enough, you have no idea whether these sequences came from the person, right? Their large intestine, their colon, the millions, billions of bacteria in there, the billions of fungus, and anything else, food particles, whatever else that has genetic material. You have no idea whether these are actually, these genetic pieces are from which one of those. But if they're, for whatever reason, they want to say these ones are viruses, they can find certain sequences which are found more often in people with colorectal cancer. They can make those into the genome representing a virus. Therefore, viruses play a role in the etiology of colorectal cancer. The conclusion of the article: powered by deep learning and high throughput sequencing metagenomic data, Deep VirFinder significantly improved the accuracy of viral identification and will assist the study of viruses in the era of metagenomics. In other words, we found a lot more of these viruses because we found a lot more of these sequences. We have no idea where these sequences' origin is. We're going to call them viruses. We're going to say that viruses, therefore, are at least associated, and the next step, they will be said to be causative of colorectal cancer. Therefore, you will obviously need a vaccine for colorectal cancer, because these viruses, which are now associated, the same thing happened with SARS-CoV-2. They said in the beginning it was associated, and then they changed it to causative with no additional information. And so you will be able to find sequences in Alzheimer's, ADD, anxiety, runny nose, emphysema, heart disease, colorectal cancer, on and on and on, because you will always find sequences and increased of certain type of sequences when the tissue is breaking down. Obviously, these are viruses. Obviously, the only way to deal with this is to give vaccines. So this sets the stage for tens, scores, hundreds, and even thousands of different RNA and other types of vaccines to combat the new finding of the viruses which have been found with deep metagenomic sequencing. Now, just to make it add a little more flavor to this, in addition, in bin 7, so the bins are one of the viruses with a negative coefficient, whatever, in the regression model, contains sequences similar to, believe it or not, CRS phage. Remember, phage or phage is the word for virus. So what they found is this is consistent with the fact that CRS phage, CRS virus, is a highly abundant virus in the healthy gut. I mean, you can't really make this stuff up. Who would have thought that the most abundant virus, or a highly abundant virus in our healthy gut, has the illustrious name of S virus? But it does. And I think finally, another interesting one, Deep VirFinder is designed for identifying prokaryotic viruses in metagenomics, which is a mixture of genomes from prokaryotic cells and prokaryotic viruses. we note that some metagenomic samples may contain contamination of sequences from host eukaryotic genomes, such as the human genome. You think? So we note that some of these samples, which we're calling viruses, by the way, may be contaminated with sequence from such a thing as maybe you. Users who have concerns of eukaryotic contamination should first filter out eukaryotic host sequences by mapping the reads to host reference genomes before applying Deep Vir Finder. Now, the problem is there is no difference between eukaryotic, in other words, your sequences, and the microbe sequences. They're all found in both. So this is total nonsense. You cannot get rid of them except or map them, or because they're basically some from the same source. But as Deep Vir Finder may potentially misidentify these eukaryotic sequences as viral. You think, in other words, Vir Finder might say, oh, by the way, that virus there turns out to be you. But that's because our eukaryotic sequences were not included in our training data set. For more complicated reference cases where host genomes are not available, forget about that. In other words, we couldn't train anybody on this, probably because there is no way to know whether the sequences came from you or the so-called virus. And it's just another way of saying the virus doesn't exist. It's just you breaking down. And where this is going to lead is, this is going to say, here are all the viruses that make up our virome. The virome is a nonsense. It's just pieces of genetic material that have been deep sequenced like this, and nobody knows where they came from. And then they're going to say, well, 46% of your genome is actually viruses, which is nonsense, because what they're looking at is just sequencing whatever's there. They have no idea, as they say, we may potentially misidentify these eukaryotic sequences as viral because there is no technique in the world that anybody can distinguish a virus from you. The sequences all come from you, and the whole thing is going to lead to the identification of more and more viruses found in association with every single condition that we have, which is then going to go from association to causation, which is then going to go from causation to obviously the treatment for preventing Alzheimer's, because we found 6 different viruses, meaning genetic sequences, in some of the people who are breaking down because they're being poisoned. And so their brain is breaking down into sequences. Those we now call viruses because we don't need any culture or any of that stuff. We just need deep learning with deep metagenomic sequencing. And then we give you a vaccine with aluminum in it to help with your Alzheimer's. And don't bother us if something goes wrong. That, my friends, is a threat to everybody.